Today we are going to read about another topic, that is complement. So the complement is the system of hepatically synthesized plasma proteins that play a role in innate immunity and inflammation. So uh, in the complement topic there is always one word, there is MAC, that is membrane attack complex. So this is the one of the important complex that helps in the cytolysis. So the membrane attack complex defends against the gram negative bacteria, especially the gram negative bacteria. The CH50 test is also used to screen for activation of the classic complement pathway. So there are three types of activation pathway for the complement system, you have to know that. So the activation pathways of the complement are the classic, alternative and lecithin induced. So in the classic there is involvement of immunoglobulin G or immunoglobulin M, whereas in the alternative there is the involvement of the microbe surface molecule, whereas in the lecithin there is involvement of mannose or other sugars on the microbe surface. Now let us discuss what are the various functions of those protein in the complement system. The C3b especially functions in the opsonization of the bacteria, whereas the C3a, C3, C4a and C5a involves in the anaphylaxis. The C5a helps in the neutrophil chemotaxis. And previously I have told you that the membrane attack complex defend against the gram negative bacteria that acts as the cytolysis of the infected bacteria. The membrane attack complex is C5b to 9, that means C5b, C6b, C7b, C8b and C9b are the membrane attack complex that enhance the cytolysis of the infected cell. The various opsonins are C3b and immunoglobulin G. They enhances the phagocytosis by the opsonization. The C3b also helps to clear the immune complexes. Now let us discuss what are the various inhibitors of the complement. The decay accelerating factors, that is DAF, also known as CD55, clusters of differentiation 55, and the C1 esterase inhibitor helps to prevent the complement activation on the self cells, that means RBC. In the RBC, that means you have to know that the inhibitors are the decay accelerating factor and C1 esterase inhibitor. They help to prevent the complement activation. So sequentially we are going to read about various activation pathway that are classic pathway, alternative pathway and lecithin activation pathway. Again, I'm going to say the classic pathway involves the immunoglobulin G or immunoglobulin M. That means the classic pathway is mediated by immunoglobulin G or immunoglobulin M, that is antibody or antigen mediated, whereas the alternative pathway involves the microbe surface of the molecule and the lecithin pathway involves the mannose or other sugars on the microbe surface. So we'll discuss this classic, alternative and lecithin pathway in detail one by one. Now moving towards various activation pathways of the complement, again I am going to emphasis on those three activation pathways that are alternative, lecithin and classic pathway. In the classic pathway, as I have already told, that there is involvement of the IgG or IgM, that means there is antigen antibody complexes is formed. And in the alternative pathway there is the involvement of the microbe surface molecule, and in the lecithin pathway there is mannose or other sugars on the microbe surface. So that helps in the activation of the complement system. Now let us discuss one by one, that is alternative, lecithin and classical pathway of the complement activation. So first of all you have to know that the complement has the various protein complexes like C1, C2, C3, C4, C5, C6, C7, C8 and C9, but they come in sequentially. But the C2, there is little bit like alteration in the C4 and C2, that means the C4 comes before the C2, and ultimately in the activation pathway, the ultimate complement protein that we need is the C3b. So the ultimate complement protein that we need is the C3b. So once we obtain the complement protein, that is C3b, then there will be the activation of the complement system. So now first of all, let us discuss about the alternative pathway. So the alternative pathway, that is spontaneous and the microbial surfaces are involved, which amplifies the generation of the C3b. So now what happens is that the C3 is automatically converted to C3b, and the protein B is converted to BB by the help of the protein D. So now we have the protein complex, that is C3BBB. So this protein complex C3BBB is also called as the C3 convertase. So C3 convertase, what does is that it helps to convert the C3 to C3b with the generation of the C3A. So now we have the C3b molecule from the alternative pathway. Now let us discuss about the lecithin pathway. So that means here what happens, there are C1 like complexes. So the C1 like complexes is already present, and the microbial surface, if they have mannose, and they activate C1 like complexes, and there is conversion of the C4 to C4b by releasing one C4A, and there is formation of the C4b and C2b. That means there is formation of the C4b2b, that is the C3 convertase is formed from the lecithin. And the C3 convertase helps to convert C3 to C3b, thereby forming the C3b. Now in the classical pathway, there is the involvement of the antigen antibody complexes. That means we have the C1 complement protein, and the C1 complement protein converts the C2 into C2b. So this same C1 also converts the C4 into C4b. That means we have the C4b from the lecithin pathway, and the from the classical pathway we have C2b. And also the C1 can convert C4 to C4b in the both the lecithin pathway as well as in the classical pathway. So from both lecithin and classical pathway we have C4b2b. That means we have C3 convertase that convert the C3 to C3b, thereby removing generating the C3A. So from all the three activation pathway, the alternative, lecithin, and the classical activation pathway, we have the protein molecule, that is C3b. And from the alternative pathway we have the C3BBB previously, which is known as the C3 convertase, and now we have C3b. So combination of this C3BBB with C3b, we have the C3BBB and 3b, that is known as the C5 convertase, that is formed from the alternative pathway. And this C5 convertase helps to convert C5 to C5b, and with the help of the C6 to C9, there is formation of the membrane attack complex from the alternative pathway. And we have the common formation of the C3b from the lecithin and classical pathway. What happens is that from the C3b we have C4b2b from the lecithin and the classical pathway, and adding the C3b we have C4b2b and 3b, that is the C5 convertase that is obtained from the lecithin and classical pathway. And this C4b2b 3b, that is the C5 convertase, helps to convert C5 to C5b, and again with the incorporation of the C6 to C9, that is C6, C7, C8 and C9, there is formation of the membrane attack complex, that is C5b to C9, and it helps to lysis of the various cells that are infected with the bacteria, and it increases the cytotoxicity. The membrane attack cells increases the lysis of the cell, and it also increases the cytotoxicity. So again I am going to describe, briefly revise the various steps that I have explained in this alternative, lecithin and classical pathway. So all this classical pathway, all this pathway lead to the formation of the C3b. After the formation, after we obtain the C3b, we obtain the C5 convertase from the alternative lecithin and classical pathway. The alternative pathway have their own spontaneous conversion. That means they have their own C3 convertase. The C3 convertase in the alternative pathway is C3bBB, and after the incorporation of the C3b, there is formation of the C5 convertase, that is C3b, BB and 3B. So this C5 convertase helps to convert C5 to C5b, thereby forming the membrane attack complex. But whereas in the lecithin and the classical pathway, the C3 convertase is C4b2b, and after the incorporation of the C3b, there is formation of the C5 convertase, that is C4b, 2b and 3b. So this is the C5 convertase of the lecithin and the classical pathway. This helps to convert the C5 to C5b, and the membrane attack complex is formed, thereby increases the lysis of the bacteria infected cell and increase the cytotoxicity. So I hope you understood this complement system and various activation pathway. Thank you.